Okay. I want to just do a review of the discussion that Anthony Maurice had with the James Torres question and answer session. This is a very significant event for us, in the creationist community, and that we have a scientist actually asking our questions, the questions that we have, to the non-believing community or skeptic community, whatever you want to call them, and because it's just not making any sense at all. So let's just go ahead and watch this conversation, and we'll see what the problems are from a scientist perspective, that there is no possible natural cause for for natural things, of course. Nothing can make itself, nothing can direct itself, nothing can order itself. Nature can't make nature, matter can't make matter, and it can't program itself either. So here we go. Let me go ahead and be quiet.
All right, guys, welcome. Thank you so much for being here. The day has arrived, and we do have a beautiful little interview here with Dr. Tour himself. Tony, thank you so much for being here and arranging this. Let's go and bring him in, because he is a busy man, and he's given us a little bit of his time to be here. Sir, it is an honor. Thank you so much for joining us today.
Thank you.
All right, guys, welcome. Thank you so much for being here. The day has arrived, and we do have a beautiful little interview here with Dr. Tour himself.
Sorry, I have my thing playing. Sorry, go ahead.
Technical difficulty.
Dr. Tour, thank you. It's an honor for coming. We can jump right into some of the problems with abiogenesis, the RNA world, metabolism first, protocells, all plagued with problems. We just would really like to have your expertise on this.
Right. Yes. So at every stage, there's a problem with origin of life. You have to make the 4 classes of molecules, and it's not known how to make them, and so that's one problem. And then so once you have to make the amino acids, and once you've made the amino acids, people will say, well, we've made amino acids in the Miller-Urey experiment 60 years ago. The problem with that is that they were all made racemic, so we don't know how to make them in a chiral fashion. Moreover, they have to be hooked together. I mean, they're not proteins until you get them hooked together, and then and it's not easy to hook them together. In fact, if you look at a DNA synthesizer, you have many blocking and unblocking and protection steps. So nobody knows how to hook those together in anything at all similar to a prebiotic environment. And then even just hooking them together doesn't solve the problem. You have to have a specific order. It's not just some random a string of amino acids, and you have 20 different amino acids, so you have so many different ways of combining these together, and if it's not right, it doesn't work. All right, and then you have the nucleotides, and the nucleotide is a base. It's a nucleobase, and then it has to be hooked to a sugar, and that sugar has to be hooked to a phosphate. Yeah. Nobody knows how to do that in a prebiotic-like environment. People can do it in the laboratory, but in a prebiotic environment, it's very hard. And then nobody knows where that sugar came from. Right. Then if you want to make the sugars or the saccharides, that's a whole nother problem. Those are probably the hardest thing to think about synthesizing. So no prebiotic chemist has ever tried to tackle those, because they are really, really hard to make. And you need lots of blocking and unblocking chemistry. And then if the order is wrong, none of that works. And these are very important for signaling pathways across a cell. And then even the lipids from which you build the protocells are not simple. You have a diacyl lipid. It has to be diacyl, not monoacyl, or else it destabilizes the lipid bilayer. And all of those are chiral. And so nobody knows how those are easily made. So that's just the basic building blocks. You want me to go to the next phase? I mean,
sure. Could you just give us some of these manipulated conditions, you've talked about this before, and some of the chemical kits that chemists have to order just to get the experiments going with all these purified reagents and chemically stabilized clays and all this stuff has to be prepared beforehand just to get the experiment going.
Yeah, but even then it doesn't work. I mean, the way chemists do this very selectively, what happens is biologists buy these chemistry kits that allow them, that are made by chemists that allow them to mix a few things together and it works. So they put it in their DNA synthesizer and it works.
Right.
Not knowing how much went into building those intermediates and then selectively protecting them. And generally, those intermediates are not made ab initio, not made from the beginning. They're generally extracted from natural sources, and then they'll put a particular blocking group on it or something. So many of these amino acids are just extracted from natural sources. It's easier to do that. And the carbohydrates the same way. I mean, carbohydrate chemistry is really a bear. It's very, very hard to do this. And these lipids as well. I mean, all of this is generally extracted from natural sources and selectively protected and then put in bottles by chemists that biologists buy, and they say, it wasn't that hard to make it. Well, you had the whole thing set up for you.
Right. It was all ready to go.
Prebiotic type conditions where you just have an early Earth with just rocks and some CO2 and hydrogen cyanide and potassium chloride or sodium chloride. You know, so nobody knows how to do this.
What about we're hearing from the atheist community, well, we can't do it in large scale amounts of chemicals. What about microenvironments? If we set up smaller environments, will this make the chemistry more plausible?
No, it won't make it more plausible because still you're without the code. I mean, it's just not going to help you. And then anytime you do synthesis, yeah, you might get one step randomly done, but you have to do that over and over again, and you have to bring it down many other steps. So unless you make boatloads of stuff, you don't have enough material to bring you down the multiplicity of steps that you'd have to get to. So that doesn't solve it at all either. Plus, you have to have characterization at each step, or else you have these terrible gross mixtures. You have no separation mechanisms that are easily available to you. You might say, well, there was a selective crystallization. Okay, there was a selective crystallization. And now what? Now you have these crystals. Now those have to dissolve. You bring them on the next step. And then when there again a very selective crystallization that somehow filtered it away from all the other things that could easily form, that none of that works. And the people who understand this are the synthetic chemists. The biologists absolutely do not understand this because they've never made these molecules. This is hard chemistry. So you don't see synthetic chemists pushing these types of ideas that robustly, certainly not in venues where they're going to be challenged by somebody that says, explain that to me a little bit. Tell me more. How was that done?
What about competing materials? Like you're saying, all that has to be filtered out. You have purification processes that have to happen through each step. That's all intelligently, that's all intelligent agency actually doing that. Those aren't natural mechanisms.
Sorry for stopping here. That's a big thing that James Torr just said, is that there's nobody to question what's going on. This peer review is just an absolute farce because nobody's questioning how they're arriving at these conclusions. And all you have to do is just ask questions as to how exactly did this happen?
That would be available on the early Earth.
Right. And you know, what's interesting about this is those separations are very, very hard. And so what happens in prebiotic in laboratories that try to copy prebiotic chemistry is they'll mix things together and then they'll run what's called an HPLC, a high pressure liquid chromatography chromatogram, and they'll see all the different peaks that they have in there. And sometimes it's just so messy, they don't even try to do the separations. They say, oh, see, it's there. And then they'll buy selectively that one and bring it on to the next step or make it using some other methods. They'll never carry it on using that crazy chemistry because that would gum up the works. That would not be possible. And even they, with all the tools, don't try to separate out because even with all the tools we have for separation, still, it can be just impossible to separate that. And none of this works together.
Right. How severe is the chirality problem with the proteins and the nucleotides? This is another atheist talking point. Oh, we have this mechanism. We're so close.
Yeah. So no, they're not close at all. So you have almost all of the amino acids have a stereogenic center. So they all have a chiral form. But in a way, and so nobody's ever shown on a prebiotic earth how you're going to get those to be stereo pure. Even if you have like plain polarized light, those give you very small degrees of enhancement, not nearly what you need. But those are child's play compared to sugars. Sugars are just wild. You'll have say 5 stereo centers on there. So you can have 2 to the 5th possibilities of isomers. So you have these huge numbers, and then the way you can hook them together and the different branching patterns. So for example, it's just so complex. The nuclear base may not have a stereogenic center, but what it's coupled to, which is a sugar, a 5-membered ring sugar, that has several stereo centers on it. You had to get that sugar, and you had to selectively couple it to that particular site on the sugar. That's hard to do. And then you have to put the phosphate at the right site. And then all the fatty acids, those are chiral as well. So you have that chirality problem. But the thing about the sugars is just absolutely daunting. And that's why none of these folks working in this area has ever tried to address the sugar problem. Well, take that back. Professor Eschenmoser, 35 years ago, tried to address it, and he couldn't address it. I mean, he showed that this thing is just really hard to do. And he was just trying to make one particular type of sugar, and he was trying to make the furanose, and he got the 6-membered ring instead. But what happens is you can have so many possible different isomers, just 6 different sugars, just 6. Generally, you have oligomers of sugars, these polysaccharides, that have not just 6, they'll have 20, 30 different sugars on them. So if you just have 6, you can have, if you just take the 1 sugar D-mannose, you have over 1 trillion possibilities of how you can hook this together, because there's so many different coupling sites, and then you have the anomeric carbon. So nobody ever addresses this. We're just clueless on this, and somebody has to call people out on it. That we're nowhere close, nor do we even have a proposal on the table on how this could have happened. So
yeah, we gotta call them out. We have to call these people out, because they have no fact, no science behind them, no basis to be pushing this idea that anything can order itself, that any physical thing can program itself, because it can't even put itself into directed working order to begin with. So how is it going to program itself? You have to have the programming to make the proteins and everything. And you have to have the everything to store the information, the programming. So it's just, and this peer review is really a mess. And nobody's even questioning the evolution, which it has no basis at all. There's absolutely no basis whatsoever to evolution, any kind of evolution, because variation, adaptation, responding to the environment, epigenetics is not evolution.
it's not just that we're not close, and it just has to be tried. Nobody even knows where to begin to try.
Right. What about now Jack Shostak and Lee Cronin? I wish you would have pressed them further. They think they're coming very close with some of their RNA world experiments. Can you mention some of the problems and things that they're doing?
Yeah,
Protocells as well.
Lee was very gracious to come and to speak with me in a recorded session, and so that was actually the first person among the Origin of Life people that would actually speak with me. And I tried to be very nice because I didn't want to scare these guys off. And when he couldn't answer, I didn't press him on it. I just went on, just as I would have at a normal scientific conference. Many Christians are upset that I didn't go after him harder when he couldn't answer, that I didn't go after him harder. But my colleagues who are professional chemists, they very much appreciate what I did. I made my point by him not being able to answer these things, and I went on. And I just think that although I respect him as a scientist, I've seen movies, videos of his laboratory, and it's amazing. He has all this computer-controlled, this adds at this instant, this adds at that instant. And even with that, what he's made is just trivia, just absolutely trivia. And then what's happened is there are certain things that you have to have that to have life. But what he's done is to, rather than go and compile all of these things, he's saying, what's the fewest things that we could have to have life, and I'll shoot for that. But that's not life. It has to have all of those parameters, and even just the one or two are hard to get, and he hasn't hit on those. And so he's using what's called an autocatalytic reaction. Yeah. But autocatalysis has been around for over 100 years. That's not life. Autocatalysis is not life. It just means that what you've made, what's formed in a reaction, acts as a new catalyst to make something else. That's autocatalysis. That's routine chemistry. That's not life. Certainly there is autocatalysis as occurs in living organisms, but so does SN1 reactions and SN2 reactions and elimination reactions. All of those occur. Just because you run an SN2 reaction, does that mean that you've now getting close to doing life? No, this is one very small component. Life is crazy complex. It is a non-equilibrium system that has to constantly work, constantly expel energy to keep it in a non-equilibrium state, because natural forces are pulling on that to get it to just diffuse out from itself. And it's constantly working, and ATP is constantly working and generating this power source for the cell to remain in what's called a non-equilibrium state. Things want to move into an equilibrium state, and life functions away from that in a non-equilibrium state. We don't know how to build things like that.
What about a more?
That's hard to do. We're clueless on these sort of things.
But they would say you're talking about a modern cell. What about a primitive cell, or these proto cells? What about all that?
All right. So let's first take the first one. A primitive cell still needs a number of protein coding genes. Some have calculated you're still going to need something like 256 protein coding genes. You're still going to have that many. So you still have to have DNA for that. You still have to have RNA being formed from the DNA, and you have to then have all the enzymes working on that. Now, some have reduced that and say, well, it might only take 100 protein coding genes, but still you have to have the DNA there, because the RNA itself is not stable enough to do things by itself. And to have the code, nobody knows where the code came from. So there's never been a simple cell that some person can think of. Even a simple cell has still got to have DNA, it's still got to have RNA, it's still got to have enzymes working. We don't know how to do that. All of these pieces are unknown to us. Now, a protocell is ridiculous. It's absolutely ridiculous. A protocell is just a lipid bilayer forming a spherical vesicle. You can do that in the lab very easily. When you make salad dressing, you have made protocells by that definition. You just take oil and water, these fatty acid oils, and you water, and you shake it up, and you get these little bubbles. That's a protocell in their mind.
Garbage.
Yeah, a protocell, as they say, is an early form of a cell. It is not. A lipid bilayer, there are many different lipids within a lipid bilayer. Many different lipids. And where do you get the diacetyl lipids? Those have stereogenic centers. Where do you get those? They buy them. They buy them in a store. When you make salad dressing, that has the fatty acid diacetyl lipids, but that came from vegetables. That was extracted from vegetables, from natural sources. How do you make those in a lab? It's not easy. How do you make them in a prebiotic type chemistry lab? Nobody's done it in high fidelity with high stereogenicity. Nobody's done this. And then what happens is once you've made this, so generally what they always do is they make this homogeneous protocell. Cells have lots of different lipids, but just the lipid itself is not a protocell. You have to have proteins going through that give you these ion channels that go through. You have to have displays of many saccharides, polysaccharides, cover the surface. And if you get the polysaccharide structure wrong in any class of polysaccharides, the cell dies. all of those have to be right. Well, how did they get the code for being right? The code originally came from DNA, and then enzymes built those first to RNA, and then enzymes built those sugars, and you had to have the basic sugar building blocks. Nobody knows how they got those. And then once you build those, that they have to be displayed on the surface. So you have to then hook those to the fatty acid. You have this chain going to it. Nobody knows how to do this. This is so utterly complex. It would be more believable that you could build a factory, a regular factory that produces automobiles, than trying to think that molecules could somehow form cells. Could you see spontaneously a factory just go up like magic? Well, is the probability zero? No, it's not zero. There is some probability that a factory could form and start producing automobiles. And they'll say, well, if the probability isn't zero, it could have happened. Yeah, well, the probability isn't zero that a car manufacturing plant could form spontaneously. The probability.
Yeah, this probability thing is actually less than zero because of the fact that everything always falls apart at a much greater rate than it supposedly puts itself into directed working order. So the time you spend waiting for something to put itself into order exceeds, I mean, it far surpasses the viability of the components themselves, which always fall apart. And if people could understand that, you could understand that the probability is less than zero.
Probability is non-zero. That's true. But it's the same. It would be even harder to make a cell. It's even harder to do this with a cell because we don't have factories that can make cells. We have factories that are human built that can make automobiles. Why don't we have factories that are human built that can make cells? Because we don't know how to. That's why.
So would you say they basically manipulated a bunch of useless building blocks, and then from there, they can't even do anything?
Well, they didn't even make the building blocks. Yeah, they didn't make the building blocks. What they made is racemic. And in the few cases where it was chiral, the degree of chirality, the degree of enantiomeric excess is very, very small. It wouldn't be usable. Wouldn't be usable. So they haven't even made the building blocks, let alone the code on how to, let alone the chemistry to put those building blocks together, let alone the code on what the order should be, let alone the construction. Even if I gave them all the building blocks in complete chiral form, chirally pure form,
right?
Could they build a cell? The answer is no. They wouldn't even know how to build a cell. There's components here that are called interactomes. Nobody knows how to get these non-covalent interactions to line up like this to give this order. Nobody knows how to do that. Even if I give you a whole cell,
any environment they want,
that has just died, and I say, bring that thing back to life. Well, how did it die? I don't know. It's just dead. It was alive, and now it's dead. Just bring it back together? Everything's almost perfectly in place. Now
almost there.
I have no idea how to do it.
Yeah.
There's a spark of life that we are clueless on. We're absolutely clueless, and that's kind of the state that we're at, Tony.
Thank you so much. Absolutely. No environment really works. Oxygen, no oxygen, control temperatures, whatever they want to put it in. It's not working.
We'll give it to them. They can use whatever they want.
Whatever they want. They have high hopes for abiogenesis.
Yeah.
Thank you, sir.
Tony.
Thank you so much, Dr. Throw. We hope to have you back. A few other people want to meet you, and they would love to hear from you. We have another channel, John Maddox, Logical, Probable, Plausible, and Standing for Truth. They would love to have you on, and we would love to have you back. We are honored. Thank you so much. God bless you, sir.
God bless you, sir. Have a great day.
Yeah, this is quite the interview, to say the least. Let me see. So yeah, it's quite the interview. Hey, Iger, oops. What up, bro? Caps reversed. So yeah, that's the state that we're at. That's the state that we're at. we have scientists believing impossible things can happen, and they have no possible way of it ever happening, and yet they believe it did happen because they don't want to accept the fact that we have a creator. And because they're, of course, they don't want to be right, they want to be wrong, and they want to wallow in their misery and complain, criticize, and attack people that do want to tell the truth and be right. So it's just that's the only thing that evolution is an attack on God. Is what it boils down to. Because if you know anything at all, you're not going to be attacking God. But anyway, I wanted to just touch on also. I wanted to touch on the idea that the continents broke apart 100 years after the global flood. I mean, I've already made videos about it, but I just wanted to condense it a little bit, if possible. So let me go ahead and screen share again before I forget. I'll be talking away, talking about something I don't even have the screen share up. Okay, screen share is on. So yeah, we have the breakup of the continents, and we have the pre-flood world and the post-flood world, and we have the world made by the flood. In other words, our geology is the result of the global flood. So what we need here is, let's just say, Antarctica breakup. Map. I'm not talking about glaciers breaking up. Antarctica, well, that's not exactly what I want here. But America, Australia, and India, there we go. Now we're talking. So this is the kind of the point here. Let me see if I can go ahead and put this up here. Oh, we got Zealandia too. Basically the same thing, okay. Okay, so we'll split this one up here. So this is what basically the result of the global flood. Okay. So when the flood happened, let's just say we have the global flood, we have all the sediment deposition and everything, and then the water recedes. What are we going to have? So this, what we see here, is exactly what we have after the global flood. We have sediment layers, of course, that are going from one continent to another, containing fossils on both sides of the divide, are going through these areas. For example, we see here in Madagascar, we got the fossil going through the northern tip of Madagascar. Of course, this is just a general idea, but just to show that these fossils pass through these regions, these parts of the continent, one way or another. It might go around the other way, or might go around the outside, or whatever. But they're connecting to these continents, what are now continents, showing that they used to be together. The sediment layers used to be unbroken, going from one continent to another until the breakup of the global flood. So then we can add one more component here. We can find it. So I'll put here glacier. Okay, so the same idea is here. In this case, we have the same idea, where we have this is we had the global flood, we had the sediment deposition of the geology that we have today, and of course, as I just mentioned, that the layers go from one continent to another with fossils connecting them. So on top of these sediment layers that were deposited by the global flood, we now have ice forming. So 100 years after the global flood, you had these glaciers moving across the continents in a particular direction. So in South America, you can see the direction was north and west. Africa was north and then south in this area. India, you have the glacial markings going from south to north. Australia, we have the same, south to north. So that when the continents broke apart, of course, the striation remained. India went all the way up to where it is today, which is not far away from here. Antarctica broke apart, Australia broke apart, South America broke apart, and they separated from Africa as well. So what you have here, 100 years after the global flood, you have evidence of India being connected to Antarctica and Africa and Madagascar. And it's really very clear evidence that the breakup happened after the glaciers formed during a period 100 years after the global flood, because the glacial striations, the markings, are going from south to north, proving that they had not yet broken apart 100 years after the global flood. And of course, we have the verse in the Bible. We have the verse in the Bible, which is the reason why I believe it anyways. The main reason why I believe it is that it says in the days of Peleg, the earth was divided. And that is 100 years after the global flood. So the Bible is borne out to be true here. And to me, it's a complete, absolute confirmation of what the Bible says. And it's really not so hard to understand. we have all this evidence that there was a global flood. It was all, together. The continents had not broken apart yet, and you have the sediment layers going from one to another. And of course, there's maps of that too. There's maps of the sediment layers going from continent to continent. So we actually got the mega sequence. Okay, see if we can find a map here of the sediment layers. I had one the other day about the sediment layers. Basically, this one actually here, this one here is basically sediment layers going from one continent to another, but it's not exclusive. Not just talking about the sediment layers. But that was the first reason why, I started considering seriously whether or not the breakup was 100 years after the global flood, was because of the sediment layers that go from America to Europe and South America to Africa. So let me see. I don't know if I can get. Okay, I get a map here. I might have to look for that sediment layers, but it's basically the same thing. But it's funny how they go through all these different time periods where they think that the Earth was going through different stages from plate tectonics, and in reality, it's just the mega sequences from the global flood. So let me just go ahead and erase some of this here and see what we can find. Of course, we have the mega sequences, which is what I was talking about the other day, which was this map over here. The mega sequences stacked like pancakes on top of each other. So I don't know how that could be different stages, tectonic stages, when they're stacked like pancakes on top of each other. So that completely debunks the tectonic cause for the mega sequences stacked on top of each other. It's just total nonsense. And they go through all these mental gymnastic hoops trying to explain the geology that we have today, and failing miserably. Okay, so I'll just say America. Between America and Africa, you can see the size of these mega sequences. They cover hundreds of miles, and they're just stacked on top of each other. Each one of these is a mega sequence, these things here. And there's like 5 of them. You can count them. They're like 5 mega sequences, or 6 actually. And so you can't, the point of the hundreds of millions of years missing between the layers are just nonsense, because it's not, the explanation is a glacier, but how are you going to have a glacier in the middle of a mega sequence? It's just total, an obvious effort trying to make excuses for the evidence that we see. Now, somehow I'm not getting the mega sequence. Well, I'm getting this one here. It does show some of the layers going across, I think. Okay, you have the mega sequences, and of course the mega sequences are across the world: South America, Africa, North America. You have the same mega sequences: the Salk, the Tippecanoe, the Kaskaskia, Kaskaskia, and they're really all over the world, these mega sequences. And they're stacked like pancakes, so that means and proves that plate tectonics is not the reason why they're there. Okay, there is a map about the sediment layers, but I guess I'm having trouble finding it. Sediment layers. Okay. So whenever you do see these time periods of Pangean, Gondwana, and all this stuff, they're trying to say that tectonic activity is the cause of the mega sequences that we see, which is, again, total nonsense. Because the mega sequences are stacked on top of each other. Here's Chinese sediment layers, which are basically a bunch of mud layers, because that's what sediment layers are. They're mud layers. Okay. See what we find here, if anything. Multi-proxy database. No, it's temperature. Don't want temperature. Okay, here we go. This might have something here. Okay. Still talking about temperature. But the reason why they're connecting everything here is because everything was connected because of the sediment layers that go back and forth between the continents, that connect them. For example, they got subtropical climate here. that connects this. Well, this is of course fossil evidence that's connecting, but it would be the same sediment layers. Same thing with North America and Europe. They're saying subtropical and arid climates. Okay, southern, just talking about cool and cold. Because as soon as this land area was made by the global flood with the mega sequences stacked on top of each other, this area in the south became cold, very cold, and with glaciers. Even South America, Africa, Madagascar, and India, and then Australia, southern tip of Australia. Okay, so they were connected because they had glacial markings going from south to north, and then they broke apart 100 years after the global flood. Now I'm looking specifically for sediment layers, and I'm not sure. Okay, they're always talking about plate tectonics, trying to say plate tectonics is what caused the mega sequences, but the mega sequences are the result of the global flood, because that's the only way you can have a mega sequence. Sediment layers stacked like on top of each other like pancakes. And it's really obvious evidence of the global flood. Okay, so I guess that's about, I guess I'm not finding the sediment layers for that. But the other maps do kind of give an idea of what's going on there. Let me see. Bag is here, or was here. But yeah, point being that this is a relatively current event, the breakup of the continents. So it would be in the timeline if the arc landed somewhere around 4,400 or 4,500 years ago, then that means that the breakup happened around 4,400 years ago or 4,300 years ago. And as I mentioned in the previous discussion, is that it cut the lifespan in half a second time after the global flood cut the lifespan in half. The event of the breakup of the continents cut the lifespan in half a second time. And it's hard to believe, but that's what the evidence seems to show. Okay, the White Cliffs of Dover, that's chalk beds. I think they extend across continents as well. Understanding how the flood sediment record was formed. See, I was surprised to see that creationists weren't able to make a determination as to which sediment layers are from the global flood. And the reality is quite clear to me that everything above the great unconformity is from the global flood, and everything below it is pre-flood sediment layers. The reason why it was hard for them to determine that is because they're not accepting the verse in the Bible where it says the earth was divided in the days of Peleg. And if they had accepted the fact that the earth divided in the days of Peleg, it wouldn't be a problem. But since they don't believe the earth broke apart after the global flood, they think it broke up before. They're trying to figure out what is from the global flood, what is not. And what is pre-flood, what is post-flood layers. So they're going back and forth between pre-flood and post-flood when all the sediment layers above the great unconformity are post-flood because they broke apart 100 years after the global flood. It's really, really simple to understand. Okay. By way of review, by way of reminder, in case people are not sure, we have the great unconformity right here, this line right here. Everything above it is post-flood. Everything below it is pre-flood. So we have the Unkar and Chunkar groups, which are sort of mega sequences. These are sediment layers that don't have any fossils to speak of in them. The upper layers have like maybe microbes or something, which is expected. But there's no fossil in there's no fossils in these layers because obviously fossils were made by the global flood. So all the fossils are above this line here, above this great unconformity line. So this great unconformity line is everything that happened as a result of the global flood. Everything below it is pre-flood. And if you follow this line here, this great unconformity line, which is relatively flat plane, it's going to meet up with this igneous rock layer here. So these pre-flood layers are at a slant. Here, slight slant, you can even almost see it. And if you follow this line over this way, it's going to meet up with this igneous rock, so that everything below this pre-flood, this great unconformity from the global flood is basically igneous rock over here. So in places you'll have some sediment layers that are pre-flood sediment layers below the great unconformity. In other places it's just igneous rock, which is, of course, there's no history of life in the igneous rock because it's metamorphic rock, granite. But people, it's amazing to me how people will say, oh, the global flood was this proof. And it's like, it's like I'm talking to a person that had no education at all. Because all of geology above the great unconformity is the result of the global flood. And of course, this, from this peak here down to this bottom, is like a mile. And if you go up in the hills, the foothills, beyond this Grand Canyon, there's additional layers on top of these layers. This is just the beginning. This is just the beginning of the sediment layers deposited by the global flood. On top of this, there's many more layers on top. And they're all, everything above this line here is from the global flood. Everything below it is not. So these sediment layers here are what extend between the continents, go from South America to Africa, go from North America to Europe. These sediment layers above the Great Unconformity. And that's what made me consider the prospect or the idea that the breakup was, like the Bible says, in the days of Peleg, 100 years after the global flood. And it seems to be borne out very, very clear to me, and I've not found a bit of contrary evidence. Okay, so we got sediment layers, mega sequence. Put Australia in here. See if this has sediment layers. There's not really sediment layers. Yeah, it's Laurisa and Gondwana, or Laurasia, Gondwana. There you go. But this tectonic things are trying to explain how they got the mega sequences stacked on top of each other, and they basically fail to explain that. But they're trying, basically what they're trying to say is that the continents were like merry-go-rounds going up and down below the sea level and depositing sediments. That's trying to say, basically that's how the mega sequences got deposited. So you have a merry-go-round of the continents going up and down below the sea level. And that's their explanation of how the sediment layers got there, I guess, apparently. But of course, there's hundreds of millions of years missing between sediment layers within a mega sequence. Which is not possible. Okay. Let me geography. Let's see, sediment layers. Geography. Geology. Okay, geology. This is what we need. Here we go. I think this is better, maybe. but it's not really showing the sediment layers, the mega sequences. The problem, if you're not showing the mega sequences, you're not aware of what is going on. What's this? Highland, lowland, desert, shallow ocean, deep ocean. Well, we can look at this one here. Oh, we can't. It's Pinterest. Pinterest. Oh, well. Anyways, I'm just going to have to use this one here as my geology. Okay, so these animals are pre-flood. They were buried in the sediments connecting all these continents. Then there was glacial ice deposited because this is basically the South Pole. There was glacial ice markings going from south to north in India, proving that on top of the sediment layers, proving that it was still down here after the global flood. So what we have is that the animals made it to South America. We have examples of animals that are the same animal both in Africa and South America. So they had time to get where they were going. The jaguar is the same thing as a leopard. The greater grison is the same thing as a honey badger. The aardvark is the same thing as the African ant bear. And you have all the unique animals in South America because they were able to reach South America 100 years after the global flood. So that's a confirmation because the people didn't start moving. The people never made it to South America from Africa because they were still trying to build the Tower of Babel 100 years after the global flood. And it was the breakup of the continents that caused all the families to isolate into family groups so that they began speaking their own languages. And then they started moving out into Africa, but it was too late because it had already broken apart. South America had already broken apart from Africa, so they were not able to go from Africa to South America the way the jaguar and the greater grison and the aardvark did. Of course, so you have all the animals unique to South America. You have all the animals unique to Madagascar. You have all the animals unique to Australia. No problem with the biblical timeline of the earth breaking apart, the earth being divided in the days of Peleg, 100 years after the global flood. So all of the objections that the atheists had are resolved by that one little verse in the Bible that it says the earth was divided in the days of Peleg, which also happened to cut in half a second time the lifespan of people living on earth. So it was a huge traumatic event comparable to the global flood. And I don't know why I'm the only one who is able to see this so far. So I have my work cut out for me to try to show what the evidence is, and maybe people will go ahead and align themselves with what the evidence is showing us, and maybe they won't. But I just think it's funny that all of this evidence is lining up. The glacial ice, the fossils, the sediment layers, the animals between the continents, the animals where they made it to. Everything is explained by the breakup of the Earth 100 years after the global flood. And it makes a lot more sense, and maybe the atheists will pay more attention to us if we explain it the right way. Geoscience, here's some geoscience. It's technothermo, something or other. Now we're getting closer. Here we go. What's this? Trying to see what this is. All right, let's look, see what this map is saying here. Okay, we got the Amazonian Craton, Congo Craton. Here we go. We got something crossing between South America and Africa there. And it's a well-known fact that the geology connects these areas. The geology connects them, the fossils connect them, the glacial striations connect them. So we have multiple lines of evidence of them being connected. It's not just an idea. You can see here, this is going connecting the continents here. It's just a matter of when. And the Bible has it that they were still connected 100 years after the global flood. Until the days of Peleg, when the earth was divided. So yeah, it's very, very interesting. And of course we can see this, that South America, Antarctica, and Australia were all connected, and even probably New Zealand. Yeah, so these are different sediment layers here, actually, and they do connect these regions here. Got the Precambrian, what they call the Precambrian. This is below the Great Unconformity, the Precambrian. So we have Precambrian going from South America to Africa, connecting them together. And Everything else is post flood because it's above the great unconformity. Okay, so see, they're trying to say that it was the different time periods that the continents went up and down, forming the different sediment layers. But the problem with that is that they're just consecutive water deposited sediment layers all the way down, all the way through with no soil evidence. And they think it's a measure of time when they're all deposited in rapid succession the same year. Yeah, what they need to do is show the mega sequences a little bit better. Okay, that's about it for now. Unless we talk about show the Y chromosome. I guess I could show the Mtd and Y chromosome and the concentration of people that dispersed from the Middle East 100 years. 100 years after the global flood when they dispersed. Okay, so we got DNA. Okay, let me just. So we know we know a lot of the Y chromosome, but let's see if we can find the mtDNA chart here. This is the Y chromosome. But it does show something interesting. It does show the fact that everyone is concentrated. The majority of the people are concentrated in this area of the cradle of civilization, in this region here. This is the cradle of civilization right here. So you would expect that the majority of people would be represented here. And what do you know? Lo and behold, the majority of people are here, and they spread out all over the world like spokes of a wheel, with different families going in different directions. So this is one family here went this way. This is another family that went this way. Another one went this way. Another one went this way. Another one went this way. We have different families going different directions from this area here. Not this area here. Not Addis Ababa, Ethiopia, where Dan was trying to say. But from this area, right? This is DNA evidence. It doesn't lie. You can't tell me that there's more variety here than here. It's not true, Dan. Okay, so I need to find an mtDNA pie chart, which have to go back up here. Okay, I'll have to put mtDNA here. And this is not mtDNA. This is mtDNA. And it's not a good one. So let me find another one. Try this one here. Okay. mtDNA. Well, it's very clear. So we see the same thing, right? Majority of mtDNA. Of course, mtDNA is the maternal line of mother to daughter maternal line. And we see a highest concentration here in the Uzbekistan area. There is a high concentration here, by the way, too. But high concentration does not equate to more ancient ancestry or line. It's not genetic distance. You have to go to genetic distance for that. And the genetic distance chart shows that everybody has their own tree. So this, by way of review, is what the trees are. You have the L1, 2, and 3. This is the would be the wife of Ham, which may have been Egyptus. The wife of Ham is the maternal line of all Africans here. So it's this purple color here, the yellow color here, and the whatever color here, this color here. The I don't know what color that is. It's a cream, some kind of color, whatever it is. Okay, so then what it is the L. Keep in mind L, M, and N. So the L is the maternal line of the African descendants, the African women, which are nowhere else in the world for some strange reason. And the M would be all these. M, the C, Z, D, G, E, Q. That would be all the M's. Which see. Okay, so yeah, because this would be probably this M would be probably the wife of Japheth, which would be all the Asian types peoples. And the ends would be all the Semitic maternal lines. Tentatively. Okay, but then again, we do have a lot of concentration here in Addis Ababa, Ethiopia, from all the trade here. This Middle East, of course, and Egypt was a center of civilization for a long time too. So that's why you have all the diversity here, and we all came from here, this area here. Uzbekistan here has like the highest diversity there is. And of course, again, everybody spreads out in different directions. See this H? The Europeans, maternal line went this way. The red, which would be that's another Semitic line. But it'd be another family tree went this way. So you have this family tree, this family tree. And the blue is the Japhethite. So you have a Japhethite represented here. And this M, of course, is Japhethite. So that'd be this light green. So there's a lot of Japhethite in this Asia and America region, as well as Semitic maternal line DNA. But it's interesting when people find out who they are, what their maternal lines and paternal lines are. For example, mine is H, which is European, which is also Semitic because it's after N. So one strange thing that seems to have happened is that the Semitic line has a lot of variation of maternal DNA here. For what reason, I don't know. Which is the opposite of the paternal line. The paternal Semitic line is very, very narrow, very, very limited. And somehow the Semitic men are few and the Semitic women are many, is what it appears to be to me. Okay, so this light green is Japhethite. This C is Japhethite. The light blue, which is right here, is Japhethite, with little slivers here and there, the light blue, sky blue. This D is Japhethite in Japan. This blue color goes all the way down into South America. That's Japhethite. This G color is Japhethite. This E color is Japhethite. And this Q color right here is Japhethite. Japhethite Mtd, maternal DNA. In other words, Japhethite's wife. Okay, and everything else is the Semitic wife. Shem's wife. So yes. So all these other colors here, which would be N, is Japhethite. Shemite. This, what is it? What color is this? Where is it at? Yeah, this color here is Shemite. This color here is Semitic. This color here looks like this color here, which is all Semitic. So this Eskimo population over here in Iceland, or okay, what's ES says Eskimo, yeah, Eskimo population here is almost entirely Semitic. All this in America is Semitic, this color here. Even into South America a little bit is Semitic. And this red color is also Semitic. So a huge amount of America has a Semitic maternal line DNA, Mtd DNA. And again, what is not would be the blue. And this color here is not in the American. That'll be the C color of mtDNA, not to be confused with the Y chromosome. We'll see. So that's about, so a huge amount, or a good percentage of the amount of the maternal line in the Americas is, in North America particularly, is a Semitic DNA, mtDNA. Okay, then we have this color here, which is also Semitic. Sky blue. This sky blue here. Yellow. Of course, the blue, which we mentioned. This pink up here. This P over here. So Australia has a huge amount of maternal mtDNA, whereas the Y chromosome is primarily Hamite, it seems. Well, Hamite and it's I think Japhethite too, but it's autosomally it's a lot of African autosomally, or the African family, because a lot of the African family never went to Africa, which is another story. In fact, most of this Pacific and Asian, Pacific and Australia are Africans that never even went to Africa, which disproves the out of Africa theory, because they're not represented here in Africa, anywhere in Africa. That's the Y chromosome, but the mtDNA African are exclusively in Africa and nowhere else in the world. Which would be strange for the out of Africa theory too. It's another strange thing that disproves the out of Africa theory. So we got a light bluish color here, which is up here. So this would be Semitic too, which could be from the lost tribes of Israel too, in this European area here. This could be remnant of the lost tribes of Israel, right here. This light colored here, because it also goes up here, which would be expected, which coincides with the IY chromosome. The IY chromosome is probably from the lost tribes of Israel. And of course we have the H, which is also Semitic. This H is also probably from the lost tribes of Israel too. So we have the lost tribes of Israel going into Europe here, which is probably what influenced Europeans more in this area than what was influenced over here in this area, which is Asiatic. So the Europeans are basically Japhethites with Semitic populations autosomally. And the Asians are Japhethites with more African Y chromosome and autosomal DNA in many cases. So, and it's Europeans and Asians that have the Neanderthal DNA. And they just found out recently that the Africans have Neanderthal DNA too. So they have DNA from pre-flood too, just like everybody else does, but not as much. Okay, that's about it for that. So that's the mtDNA. And just to confirm, we'll look at this map here. And you got the L0, 1, 2, and 3, where's 2's over here. 2, 3 is exclusively Africa, as I said. The M would be the Japhethite mtDNA. So the Japhethite is concentrated, a large percentage concentrated in this area, and this area. And this area here, or whatever. Hard to say because they got green here too. So, they got Americas are out of the picture? Yes, America's out of the picture. And of course, this would be the Shemite mtDNA, the wife of Shem. So it'd be all this, which is, I guess, this blue. And this area here, and this area here, which is basically what I've already stated. And of course, this is a mixture of basically everybody. So that's what it is. It's L, M, and N. It's very confusing. They gotta have a better system than that. It's really very confusing. And maybe it could end up with the Y chromosome. Oh, what happened? This is missing it's missing the scale on the bottom. But anyway. So yeah, they have the Semitic region. Semitic region would be J and I and G and H. and F is around here somewhere too, but it's F, G, H, I, and J are the Semitic Y chromosomes, which is, for some reason, is a very small population. I mean, small percentage. Then you have the K, K, L, M, N, O, P, Q, R, and S are all over here to the S. And then the people that are connected to the Kushite tree, the Africans that never went to Africa would be the C and the D, the Andamanese that went all the way through Tibet, all the way up here, through here, over here. This is not a very good map. And to Japan, so that 1/3 of the Japanese population is from the DY chromosome, which is the same family of the Kushites that never went to Africa because Nimrod was the king in the land of Shinar in Mesopotamia very early on, and all his descendants went all over the world, the CY chromosome, all the way to Africa, all the way to Micronesia, Melanesia, and Polynesia, all the way to the Americas, northern tip of South America. And the sons of Ham are the Canaanites, the Cushites, Mitzrayim, the Egyptians, and Put, the Phoenicians, who are the Berbers today. And so the DNA doesn't lie. Everybody's present and accounted for. Even the IY chromosome, the lost tribes of Israel, which is myself, me, myself, and I am. That's what I am right there. But this is not a very good map. I wonder if I could get a better map. Hold on. I'll put the chromosome in here. Okay, much better, much better. Okay, see this I1M253? That's what I am. It's my paternal line, which is very closely associated with the JY chromosome, which is the Jews and the Arabs. As you can see right here in this, here the I and the J are very closely associated. They come off the same tree. It's a different, I is a branch from the same tree as J. And that's what I am. So as I was saying before, you have the A, B, C, D, and E. Of course, they got the D and the E closely associated, and the C is the son of Cush too, for that matter. And then you got the FG, you got the F, G, H, I, and J, and then you got the K, L, M, N, O, P, Q, R, S. Those are the three family trees that all connect up back to Noah, so that everybody's present and accounted for. So that's about it. See what we got here. Anybody? Anybody? Okay. Let's see what we got. We got Iger. Even in carnivorous ape, even if that were remotely true, we would see a population genetic bottleneck in every living species in the world at the same time. We don't. DNA doesn't lie. Well, as a matter of fact, we do. And because everything came off the ark. It's the only history we have. We can't make up history that's not there. Yeah. See this? So there. See, I know we came from a 4 billion-year-old rock soup that turned into a magic frog. That somebody kissed. Turned into human. Yep. That's right. That's, it's either one thing or the other. It can't be both. Wow, look, you got the whole, look at that. Wow. You got the pudding soup there. You got the fish. You got the frog. You got the reptile, the birds, and the rat, the monkey, and the people. Wow, that's pretty good. Yeah, yeah. We came from the cradle of civilization, and that's where we spread out. 100 years after the global flood, the continents broke apart. That's when people started spreading out. That's why they never made it to South America from Africa. The people that went to Africa. The other Africans that never went to Africa went the other way and they reached South America. So there. And I can prove that with this chart here. See? See the A, B, and the E never made it to South America. See, the ones that did was the C, because they went the opposite way. They went east. The C went east instead of west. So the C never even went to Africa. The D never went to Africa. So you don't have an out of Africa theory that's worth anything. Oops. I didn't show. See, the C went all the way to Americas here because it went east instead of west, never even went to Africa, ever. The D also went up through Europe, all the way up here, the D, Paleo-Siberian, the Jomons, and the Japanese. D. They're paternal descendants from Kushite, from this Kushite, or this Andamanese that never even went to Africa, but he belongs to the same tree as the C and the Kushites, the dark Africans. They never went to Africa. They went all through Asia, all through Micronesia, Melanesia, and Polynesia, and even all the way to America. They never even went to Africa. So there. Let's see. No, I'm looking at what the evidence. We're looking at the evidence. This here is not the evidence. This is a story. This is a story that you made up. Okay. Let's see. Physics didn't work back then, so the continents splitting up didn't boil the oceans. You do realize that that water is an insulator, don't you? Okay, because, well, I mean, see, because India was connected to Antarctica and Africa and Madagascar and Australia or near there, and today it's not. So that's the history, and it does have ice markings, glacial markings going from south to north. Yo, Matt Bond, how you doing? Okay, so anything before the flood is known as prehistory, about 4,400 years ago. Yep, and we got the picture to prove it. We got the picture. Let me see. We got the mega sequence. Mega sequences to prove it. Right. Oops, I can't buy it. We got the mega sequences to prove it. See this? This is the great unconformity here. Everything above is from the global flood. Everything below is pre-flood. No fossils in these sediment layers, except for microbes. And in this lower layer, there's the Unkar and Chunkar groups. The lower layer has no fossils at all. And below this is igneous rock. There's no fossils in igneous rock. There's no history in igneous rock. And these sediment layers are from the pre-flood water world from the days of creation, right here. You could go here and touch these rocks, these layers that existed from the creation of the world. You can touch them with your hands. You can actually touch the sediment layers from before the global flood with your hands. How old is written history? Tell me what you get. I get 5,000 years old matches up with Noah's flood. Yeah, every calendar, every historical record, every genealogy only goes back to 5,000 years to Noah because Noah was born before the flood, and that adds up to 5,000 years. So even that adds up. So, so yeah, it's pretty good. It's pretty good. All the evidence is on our side, by the way. So I guess I'll conclude it here. Oh, we got Anthony Maurice. How you doing? Did a great job last night, that's for sure. I just reviewed the video. I mean the James Tour interview that you made, and then I went on a sort of a rant, but it was a long rant on not about evolution, but about the breakup of the continents. Because I believe that they were 100 years after the global flood. So, and so I had to, you know, present the evidence for that. Carnivorous, have you any nice cartoons that I can laugh at? Oh, what do you got here? What's this guy? We know of multiple supercontinents before Pangea where tectonic plates just dancing around the entire forming maw. See, that's what I explained in this video right here and now. If you rewind, is that they believe that the continents were like merry-go-rounds going up and down, depositing sediments over time, and it's really more easily explained by the mega sequences deposited by the global flood, all in 1 year. So, because they're stacked like pancakes on top of each other, you can't have millions of years between sediment layers within a mega sequence that was deposited by water. So, no, your belief in Pangea or whatever it was, your belief in Gondwana or whatever it was, Laurasia or whatever it was, you're just trying to explain away the fact of known history of the global flood. Tor said, no such thing as primitive cells. All cells would require complex proteins. Yeah, it's highly recommended that you watch the interview that Anthony Maurice had with James Tor, because that really seals the deal as far as the fact that no biological mechanism can ever form on its own. It's just total absurdities. Yeah. And then a monkey went back and turned into a whale, right? Total nonsense. So that the earth broke apart 100 years after the global flood, the Bible actually says it did. Question, what came first, whale or dolphin, according to evolution religion? Yeah, they have a common ancestor with a monkey. See, right here. Right there. Common ancestor is a monkey. See that? Never read anything for the evolution religion. Evolution is a religion. Does Carnivorous Ape know who James Bond is? I mean, Matt Bond? Guess not. So yeah, it is a belief. It's a baseless belief. See, it's a baseless belief. Atheists have imaginary mechanisms for how proteins could have formed out of a prebiotic soup, which otherwise known as rock soup. Hello, James Bond. I mean Matt Bond. Yeah, this prebiotic soup. That's Iger's rockfish soup. See, and the common ancestor of a dolphin is a monkey. See that? According to the faith of evolution. It's the faith of evolution. Okay, so which one of them came first then? They evolved from a pig. That's crazy. That's crazy. Evolution is crazy. I don't know. I don't know why anyone believes in evolution. You have to be out of your mind and batty. You have to be batty to believe in evolution. Just out of your mind, completely out of your mind to believe in evolution. Because it's not what the evidence shows. The evidence shows that we have a creator who is much more intelligent than we could ever conceive of. And if you look at the biological mechanisms, which James Tour describes, and also Otangelo, the mechanisms that Otangelo is describing is just mind-boggling. You have, I mean, just go look at it. Just go look at it. The mechanisms. You have the DNA cutting itself apart and resplicing itself. So there's mechanisms that cut the DNA, untangle it, and resplice itself together again. It's just, you know, it's a intelligence that's far superior to our. It's this nanotechnology. It's nanomachines, nanotechnology, and it's not just a chemical reaction. It uses chemical reactions that have to work. They have to be what they are to work as they do, but it uses those chemical reactions by way of being directed, having been directed, by way of signaling, by way of commands to 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 have all these working mechanisms, working nanomachines for a function, for the purpose of the existence of life. And of course, they don't even know what life is. Even if you have all the mechanisms in place, they don't even know what sparks the life or what life is. Yeah. Our written history shows we came from a creator. Yeah, that is more logical. Yeah, the Bible has information from before the global flood, which is more than 5,000 years ago. A dragon is another word for a dinosaur. Yep. So as soon as everybody gets an education, they'll find out that life can't make itself and there is no evolution because you can't use unrelated mistakes to write vast amounts of programming that's not there. And it doesn't explain how the information got there in the first place. You have to have a creator that knows everything, that made everything to work, because there's 21 elements that are essential to life, without which, if you're missing one of them, you don't have life. And they're the most common elements that we have, plus an additional amount, equal amount, that are beneficial. So even the elements of the universe, the most common elements of the universe, are essential for life. They're working parts, essential working parts that we consist of, that make our existence possible, of course, which is also made possible by dilated time and distance, which is made possible by the amount of matter in the universe. And now they're coming to realize that all this dark matter they're talking about, it's probably just black holes, all over the universe. So, it's another furada, like they say in Portuguese. It's another mistake by um, evolutionary scientists, naturalistic believers in fish soup. Rockfish soup. Same thing. A serpent is a dragon. A dragon is a serpent. Same thing. The priest. The priests of evolution. Yeah, go talk to your priest of evolution. See if they can tell you where the information came from. Go to your priests and ask them where the information came from. And the working parts. And the simultaneous formation of all the working mechanisms that you have to have in place to contain the information. You can't have information stored without some kind of a mechanism to contain the information, like a computer or like DNA or like sugars or whatever it is that has the information. I mean, there's so much information they don't even know where it's coming from. And they're learning more and more, and the more they find out, the more evolution will be disproved. It's like you guys are you're in a pan, in a frying pan filled with water. You're a frog in a frying pan filled with water that's being slowly heated up, and you don't know that you're going to be boiled. By the time all the information comes out, you're going to be boiling. It's going to be too late because you didn't have any sense to jump out of the frying pan while you had a chance. Yeah. FYI, Santa Claus is fake too. Well, I'm getting a little bit tired of doing this right now, so I guess I'll just end it right here. I don't mean to cut everybody off, but hope everyone has a nice day and everybody learns from what the evidence is and what the facts are and not get too offended by what the evidence is. Because the evidence proves you have a maker. And only your maker can remake you again. Nobody else can.